Thank you very much. I hope that you guys enjoyed the conference as much as I do so far. So I will be talking about like Python's life of brain, and it's kind of like a mysterious title. So I figured that I will show you some like a highlights or like a keywords, if you will, to explain you better what I will be talking about. So I'll be talking about like brains, but like more widely, I will tell you about the applications of Python to neuroscientists, to neuroscience. Python, I mean, it's kind of obvious. Why is it even here, right? But I'll be putting a lot of like a Monty Python quotes, so kind of like a catch them all situation. You can play it. I'll be talking about fruit flies. This is kind of weird, but yeah, you'll learn why in a second. Finally, I'll tell you a little bit about those, like a fancy neuroimaging devices that let us to have a look what is happening inside the human brain. Modeling, but not kind of Victoria's Secret kind of style of modeling, but it'll be rather like a computer numerical simulation. So the goal is that towards the end of this talk, you will be able to create your own brain in Jupyter Notebook. How exciting is that? But before we move on, as this is my first PyCon Italia, let me briefly introduce myself. So my name is Dominik Krzeminski. I know that my last name is kind of a nightmare, but it's actually pronounceable if you like break it down like this. Krzeminski is like close enough, so like quick lesson of Polish for you. Who am I? I'm a neuroscientist. I work as a research associate at the University of Cambridge, but my background is in data science. So I used to work for like a larger, small companies. Now I run my own small consulting company as well. But most of all, I'm open source enthusiast, and I started contributing to open source Python packages at the very early days of my undergrad studies. So we can find my commits in packages like scikit-learn, NumPy, and so on. But I try to also promote the like a good programming practices among like a wider research community. So we can find like more about like what I do on my website or Twitter. And before I show you any of code, I want to also address like one fundamental question, which is why Python is actually useful in the neuroscience. So first of all, we as neuroscientists, we deal with quite large volumes of data, and you may ask like what kind of like volumes we are talking about. So it really depends on the resolution, right? So when we are interested in analyzing just like human brains, it might be like gigabytes of data, but then we record like multiple sessions, and very often we record it from multiple subjects to get like any meaningful statistics, right? Then when we go down with the resolution to like electron microscopy level, so like nanometer scale, the like a size of that data rises to like terabytes, hundreds of terabytes, or even like petabyte scale. But and apart from that, we also run a lot of computer simulations to test our hypothesis in silico first before we start torturing animals and human subjects. But those two first arguments I actually threw for like any, like any technology, any programming language. So what is like specific about like Python that neuroscientists like it? So first of all, I'm sure that you will all agree that Python has fairly like a gentle learning curve. And in neuroscience, a lot of people are not skilled and trained software engineer. They come from various backgrounds like psychology, biology, physiology, etc. Then there's MATLAB. So for those of you who don't know, this is like a commercial software with its own scripting language that allows people to perform some like numerical analysis. And its syntax is, let's say, like quite similar to Python, so the transition is fairly easy. And finally, some of my neuroscientist friends, they really like the code sharing extensions to Python, like Jupyter Notebooks, Google Colab, etc., etc. So when it comes to numbers, I think that the popularity of technologies that we use in neurosciences looks pretty much like this. And as you may notice, there's something interesting going on over here. I mentioned you that this MATLAB used to be quite popular, but I think that we are living in quite interesting times where something like this happens. Which is good for two reasons. So first of all, like MATLAB is commercial software. So when we train our students with it, when they leave the lab or like university, they can't really continue with their analysis unless they buy like very expensive license. And I also think that MATLAB teaches you some like about programming practices, but let's not go any deeper into that. So now very quick, like a brain anatomy, like a one-on-one. So the fundamental building block of the brain is neurons. Neurons are those like a wacky train-like structures that look pretty much like this. So we have dendrites on the top that gather information from presynaptic neurons, and that information reaches soma, and then we have this like a one little trunk, which we call axon, that passes information like forward to the next layer. Finally, all those neurons are organized, at least in the neurocortex, in those columns that are heavily connected, and they consist of like around like 200 neurons. And finally, looking more like holistically, we have the whole brain, which looks a little bit like a walnut and has almost like 1,400 cubic centimeters in volume. It weighs around 1.5 kilograms, and it consists of like 16 billion neurons, the human brain, right? And each neuron creates 10,000 connections. So you can like appreciate how difficult problem we are dealing with. So I guess that it comes as a no surprise that for those like a three different scales, we also use like a different neuroimaging technique to analyze our data. So to look at the specific synaptic connections or individual neurons, we use electron microscopy. To analyze like, again, neurons or like a group of neurons, we can use like a light level microscopy or local field potentials recording. But when we don't really want to inject anything into brain, we still can see some aspects of its activity by using EEG, MEG, and fMRI devices. So following that, actually when it comes to software, again, there is like a whole variety of like a Python packages that let you analyze that data on those like a various scales of analysis. And this is how I organized my demo part, basically. So we'll start from this like a low level from individual neurons, and we'll reach the whole brain. So the whole code of that presentation is over here. I actually, for convenience, posted this like a QR code, so you may scan it and like follow along. But if you'd like to repeat all that back at home, I will post that link later at the Discord or at the end of my presentation as well. So yeah, let's kick off with the first package, which is NAVIS, which stands for Neuron Analysis and Visualization. So this is something that we develop actually in my lab in Cambridge. NAVIS works really well with like IPython notebooks. It has like, it supports some parallelization of the analysis, and it analyzes primarily morphology and connectivity of individual neurons. As you can see from like this graph, it has this kind of like a modular structure where you can connect to it various extensions and different data sets. I highlighted one here specifically, which is a package that I wrote recently. It's called like Navis ML. It is basically interface between Navis and scikit-learn that lets you perform some like machine learning analysis on neurons. So let's go to our first demo. So this is basically the structure that you should see in the repository. There is some Conda environment for your convenience, so you can just create new environment with all the backend libraries installed. So if we look at the Navis specifically, let's import the library. And for our convenience, there's some like example neurons included. So we are reading our first neuron to the memory. And if we display and we call the cell like this, we will get this like metadata that describes like various features of that neuron, including how long it is and some others as well. But you know, this is kind of boring. So actually, let's have a look how this neuron actually looks. And when we use this like a plot 3D method, we will see this interactive plot that is actually run on Plotly with our like a first neuron. And again, if you remember this like a diagram that I showed you, we have like a soma, dendrites, and this like a long axon that passes information forward. So what is better than single neuron? Well, 3 neurons. And in Navis, we have this like a convenience data structure, which is called like a neuron list. And again, it's like really handy because just when you call it like this, it prints out this like a pandas-like data table, the data frame with all the metadata about your neurons of interest. Finally, we can again plot those like a bunch of neurons all together. There's also some different methods for making like a static plots if you need them just, you know, like copy straight away to your like research paper. But like so far, all those neurons that we have seen so far, they sort of like a hover in the void, right? So it would be better to like position them somewhere in the brain. So those are actually fruit fly neurons, just because in our lab we primarily work on fruit flies. And for those of you who haven't seen that so far, you will see for the very first time actually fruit fly brain. So Navis also for convenience has this example volume with fruit fly brain mesh. And here you can see actually the actual length and those 3 neurons in the perspective. Cool. So just to finish up about like Navis, so this is one example of the analysis that you can do with that tool. So for example, here we identified all the neurons that form this structure that is called mushroom body, and mushroom body is responsible for like decision making and olfactory memory formation. And we identified with very simple morphological clustering 3 distinct types of that neuron within mushroom body. And in Navis, it was like literally like 20 lines of code. Okay, but neurons don't only look nice, they also like behave in very interesting way, right? So most of the neurons like gather information from like pre-synaptic connections, and then when all that information reaches some like threshold, the action potential is like sent to the output. Very often for convenience, we just like represent that as like a 1 and 0 that happens in like particular time step. So to simulate neurons like this, we can use this like Brian package, yet another reference to Monty Python. And Brian is a simulator for spiking neural networks designed to save time of the processors, but also scientists. Hence, its syntax is designed to be quite easy to learn and use. And hopefully, you will appreciate this syntax in this next demo. So I import, well, everything from Brian. I know that some people hate that syntax, but Brian provides you with a lot of like units, and there's just like way too many units to import them all one by one. And what is cool about Brian is that when you solve your like mathematical model that describes the behavior of the neuron, you can simply type in all the equations here as like a string, and Python analyzes that with some like symbolic math library behind the hood and creates your model and solves it in numerical way. So here, despite the name neuron group, we will actually create a model for like a single neuron to start with, and we'll display its voltage. So what is happening here is that neuron receives some information, it like charges up slowly, it sends some spike, and then it resets back. And then this process is repeated over and over. Again, like more interesting than like a 1 neuron is like a whole bunch of them. So this time for simplicity, I decided to simulate 100 neurons. All the equation and all the structure of the code is pretty much the same as in this previous cell, with like a one little difference that this time I record not only voltage, but also spikes. So if we run this model, we'll get this fancy looking raster plot that basically shows at what point of time which neuron sent a spike further. Okay, so with that, let's move on to Nengo, which is yet another Python library for modeling of neurons. But this time we climb in the on the abstraction layer a little bit higher. So Nengo lets us to build and deploy biologically plausible neural networks, but this time we don't really need to write any equations. This is sorted for us. So Nengo comes with this like GUI that I will showcase you in a second, but it's quite easy to exploit on the latest hardware, for example, neural processing units. So like one commercial NPU that I know that is available, it was released by Intel, it's called Loihi, And if you implement your algorithms with like a spiking neural networks, then you can achieve very good energy efficiency. So for example, you can solve the MNIST classification task, but it will consume like a 10 to 20 times less energy on those units. So now let's see example of a Nengo model. So I import all the required libraries, and here what I do, I defined this model, and within this model scope, I define the stimuli, so external signal that I will send to my neuron ensemble that consists of like 100 neurons. This time I don't really define the equation, but Nengo knows and actually uses pretty much similar neuron like abstraction simplification that I showed you on the previous demo. And now we connect stimuli to our ensemble. And if we run our GUI, we'll see this whole like experiment over here. And now I can display the values that I'm interested in. So I'll be interested in the overall value that this neuron group represents, but also in neuronal spikes. So when I run it like this, you can see that this neuron ensemble actually represents the sine function that I like defined over here, right? So that's pretty cool. So now, for example, I can change the period of that function, so slow it down a little. And when I run it again, bigger, sorry. Is it better? Cheers. So now again, I display my value and spikes. Actually, I noticed that in the previous run, it was showing only like 10 neurons out of like 100. So now, once again, you can see this time our like a sine function is slower. Cool. But I promised you at the very beginning that you will be able to build the whole brain, not only like selection of neurons, right? And in order to do that, there's this other package that is called the virtual brain, which is actually a platform for like a large-scale brain simulations. And this time, rather than defining the models of like individual neurons, what we do is we define models for this kind of like a graph structure where the nodes of that graph are actually specific brain regions. So here we define the whole behavior of like a big neuronal populations in like specific brain regions. So let's have a look how we can do that. Okay. So here, for the sake of time, I won't be discussing like every single line of code. So I create this like a convenience function where I define my simulator and the equation that actually defines the model that runs within each and every node of this network that I just like showed you. And now also I need to define the edges. So how those nodes of the graph, so how those like regions of the brain are actually connected, and this can be estimated using, for example, diffusion tensor imaging. So this is like a data-driven, but there's example of such a connection matrix here. So now I run this model, and it runs for a little while. So in the meantime, I will tell you what is happening here. And what is happening here is that I will try, for the sake of this demo, to compare the structural connectivity, so the one that is like happening, so the wiring in the brain, with functional connectivity. So how actually brain regions talk to each other. And to avoid like any bias, very often what we do, we split the signal into non-overlapping windows. We compute correlation, and then we average that out to reduce the noise. So I'm doing exactly that over here, and I can display that. And here is that comparison. So here is the structural connectivity, so the actual like wiring of the brain. This is like the whole left hemisphere, right hemisphere, and corpus callosum, which is this like a structure in the middle that connects hemispheres. And this is a little bit different simulation of how those different parts of the brain talk to each other, at least according to the model that I defined before, right? What we can also do with the virtual brain is to actually display those correlations of this like a region of interest. So here is some like a posterior region and its correlation with all the other brain areas. So let's maybe have a look at some other plot like this just for the sake of appreciating it. So here is like a correlation, again, analogously with from some like a frontal region with all the others. Okay, so last but not least, so now we have like this model of our brain. It like generates some signal, and like very often we are also interested as neuroscientists with not like modeling work, but with actually what is happening within the brains of our like a human subjects, right? But we don't really want to, like we as a subject, we wouldn't allow to inject ourselves like any electrodes to the brain. So we as scientists, we are limited to gathering some brain activity from outside our heads. And we have some tools for that. There's like fMRI, but here I will present you like EEG, which is this kind of like a swimming cup that measures the electrical activity of the brain, and also MEG device. It looks a little bit different, but basically the idea is the same with that little difference that we measure a magnetic activity of the brain. So now in this very last example, I will show you very short experiment. So again, like for convenience, the authors of the M&E package included this data set from this experiment where people were sitting in the MEG scanner and they were listening to some sounds, right? So I load the data, I do some simple pre-processing. As you can see, it's literally 2 lines of code. I pick the sensors that I'm interested in, and like this cell is basically exactly what I like told you about in the previous demo, which is like splitting like the signal into those like a window that I want to analyze them in separately. And now I can, for example, with this 1 line of code, plot the spectrum of the signal. So basically moving signal from the temporal space to the frequency space. So this is basically, I broke it down, what is the frequency load to the signal. And as you can see, there is this noticeable like a peak around like 8 hertz. We call it alpha activity, and alpha is very often associated with the focus and attention. Now, similarly, we can break that even further and visualize it, how various like frequency ranges, they have like different names in the EEG literature, like delta, theta, alpha, and so on, and what kind of like how they contribute to specific like brain areas. I'm not sure how clear is that for you at the back, but as you can see, there is like a darker shade around the temporal areas, so at the side, which is exactly the area that processes the audio information, right? So that agrees with what we were stimulating our participants with. So that's pretty much it. But you know, like there's like a even bigger selection of the packages that exist in Python. So there's NiBibel that lets you analyze fMRI data, PsychoPy, with which you can create some cognitive experiments. There's Neuralib, that is basically a competitor of the virtual brain. And finally, there is connectivity. So this is the package that I created to easy analyze the functional connectivity of the brain. But there's obviously many, many more. And to sum up, I basically want to highlight that Python becomes more and more popular programming language in the brain research. And as I like proved you that you can analyze and model many different scales of the brain. And I think that this is good because that contributes to increased reproducibility and robustness of scientific software, and hopefully towards it is a step towards the democratization of science. With that, I would like to thank you all for your attention, and I would be more than happy to answer your questions. Thank you.
Thank you very much, Dominik. Thank you all for listening. So one of the first version of the audience was about these great simulation packages. Are there, by any chance, take some human brain neurons, for example, from EEG or fMRI, in order to create the simulation of the neurons? And how are the connection between the actual data and the package that then is released to have the simulation.
So yeah, so in this like a specific like package that I showed you, the like virtual brain, the way to start it, you need to somehow estimate like the edges of that like a graph that like represents the brain. And like to do that, you actually need to use some neuroimaging to basically estimate the strength with which certain nodes of the brain connect to each other. So this is like a very data-driven process. But then to understand the function of it, you can apply like any dynamical system model like you want within the nodes of that. You can estimate your functional connectivity and basically compare how it differs between the models, but also like between the structure and the function of the brain.
Great, thank you. I think that answered the questions. And so another question that has been asked is, for your experience, for what you showed and what you had in mind, how you think to apply machine learning, and especially if there is also some other libraries that you want to suggest for that?
Yeah, so I mean, like machine learning is like really big thing in like neuroscience recently, just because for the same, very same reason that I mentioned at the beginning. So we are dealing with like a huge volumes of data, and you know, like it's impossible to like analyze it like by hand. So we definitely use some like dimensionality reduction techniques, but also like classification for the task we are doing. In fact, some of you might be interested that the author and the group behind this like MNE Python package is exactly the same group that created scikit-learn. So it becomes like a more and more popular thing in neurosciences too. Mm-hmm.
Thank you very much again. Are there any more questions for the audience? Otherwise, I will ask one. So regarding the reproducibility of the research in neuroscience, we know that it grows over time in terms of open science, but still remember some years ago that the process itself, especially for neuroscience, especially in the side of the software, has been going in the right direction. Mm-hmm. And I want to ask you if using simulation is actually accelerate this process in having a reproducible research and also leverage open science in the science of reproducibility of the article, but also of the open science, open source tools that has been used.
I think it is, and I'm kind of lucky because I work in the lab where we like really care about like a good research software practices, and we open source like pretty much like all of our work in form of, you know, like a Python and R packages. And I also like as a part of this like a software sustainability institute, I try to actually like promote those like a good research practices and like a code sharing with all the other labs.
Thank you, that's super.
A follow-up question regarding this. Do you publicly when you publish some article, do you public the code as well?
Yes.
As to reproducibility?
Yes.
Yes.
So
everything goes.
Yeah. So
the data, the code, and if you
like with data is like bigger of an issue because like with those volumes you can't really like push them to GitHub, but there's like services that do so. But like with codes, well, I mean, we keep it like open on GitHub. We maintain it regularly, and then we do like a timestamp of the like a version that we are like that we used for, you know, our work on like certain article, and we like include that specific version together with it. And I think that this is the way to go.
How many times did you have to redact or correct article based on the code?
So you mean like we got some questions regarding our code?
And then somebody actually fixed your bug, and then you got totally different results.
Well, I mean, fortunately, if anything, we never had like any critical bugs. But I mean, some of you might heard about this situation that in, you know, like fMRI research, they actually like discovered like a really fundamental bug in their like analysis software that basically invalidated around like 40,000 research papers. Like, hence, you know, like keeping that open to public. And, you know, having another pair of eyes from like a different labs is like so important. So I think it's lunch, right?
Yes, so thank you very much again.